Good evening, class. Welcome to the fourth lecture of evolution series. This is the part first of evidences of evolution. Okay, in this part, we will only cover the paleontological and comparative anatomical and morphological evidences. Let's start with paleontological evidences. Okay, basically, these evidences are the evolutionary proofs which are derived from the study of fossils which are preserved in the earth crust. Clear? As we know, all the extinct organism no longer present today. Okay, so these evidences, paleontological evidences, help us to study the order of appearance of different life forms. Clear? From simple to complex one. Then the presence of transitional forms which link major groups, okay, and also which demonstrate the descent with modification. Then these type of evidences also help us to study the role of extinction and adaptive radiation in shaping biodiversity. Clear? Now we are saying again and again the word fossil. So what is fossil? Fossils are the preserved remains, impressions or traces of organism from the geological past which are found embedded in sedimentary rocks. Basically, these fossils are the remnants of the organism which were present in the past, okay, in the form of impressions or some small traces also. Clear? Fossils can be found in the form of entire organism, okay, some particular parts also, and indirect evidence like footprints or bureaus of those extinct organisms. Clear? The process of fossilization generally involves burial, mineralization and chemical replacement which protect organic material from decomposition. Clear? Then the types of fossils. The first type is actual remains or the body fossils. These type of fossils include preserved part of organisms such as bones, teeth, shells, seeds or leaves. Okay, in very rare cases soft tissues are also preserved. Okay, it is mentioned rare because it is very difficult to find soft tissues embedded for a long time, because the bacteria or the decomposers decompose these type of soft tissues. That's why. And provide direct anatomical details for studying evolutionary morphology. Clear? Actual remains include preserved parts of organisms like bones, teeth, shells, seeds or leaves. Clear? For example, woolly mammoth carcasses preserved in Siberian ice with flesh, hair and bone intact. Here you can see a woolly mammoth which was found preserved in the Siberian ice with flesh, hair and bone intact form. Clear? The next type is petrified fossils, or we can say mineralized fossils. These are the the organic material is replaced by minerals such as silica, calcite or iron oxide. Okay, the organic material in the body of those organisms is replaced by minerals like silica, calcite or iron oxide. For example, the petrified wood, okay, the petrified wood where cell structure are preserved in stones. Here you can see, important for reconstructing fine structural details of extinct plants and animals. Okay, these type of evidences are crucial for reconstructing the structural details of the extinct plants and animals. For example, petrified wood from Arizona's Petrified Forest National Park, where trees tissues are replaced by silica. Here you can see. The next type, the third one, is molds and cast. Firstly, molds are the impressions. Molds are basically the impressions left by the organism in sediments after the body decays. Okay, let's suppose an organism died. Okay, then its body get buried under the soil, and after a long time, okay, its body leave an impression on uh, some surface like rock or soil. Okay, so what are cast formed when molds get filled with minerals, creating a three-dimensional replica. This is useful for organisms with soft or fragile bodies where actual remains are absent. For example, the ammonite shells preserved as cast in limestone. Okay, here you can see these are the ammonite shells which are preserved as cast in the limestone. Clear? The next type of fossil is impressions or compression fossils. Thin, flat imprints of plants, leaves, or soft bodied animals on rock surfaces, often seen in shale or limestone. These type of fossil preserve the external morphology but lack the internal data. Okay, this is the example, the leaf imprints of ferns and cycads from Jurassic period. Here you can see the imprints of a cycad from the Jurassic period on a stone. Next is coprolites or fossilized excreta. These are basically the feces or the excreta of animals, providing evidences of their diet and their feeding habits. Clear? Especially significant in paleontology of dinosaurs and prehistoric mammals. Clear? For example, dinosaur coprolites from the Cretaceous revealing plant fibers and bones inside. Here you can see the excreta of dinosaurs from the Cretaceous period, which shows the plant fibers and bones inside. Next are the trace fossils. Okay, trace fossils are basically indirect evidences of biological activities such as footprints, burrows, trails, nests, and feeding marks. These type of fossils reveal the behavior, locomotion, and ecological interaction of the extinct organism. Clear? This is the example dinosaur footprints in Utah in USA and trilobite burrows in Cambrian rocks. Here are the traces of dinosaur footprints, and here is the print of trilobites. Next, amber fossils. Amber fossils, also known as resin fossils. Okay, so organisms, mainly insects, spiders, or plant parts, trapped in plant resins. Okay, that hardened into amber. Okay, these organisms were trapped in the resins secreted by plants, and these resins, when become hard, converted into amber. These type of fossils provide exceptional 3D preservation of delicate structure. Okay, here you can see a spider is preserved inside this resinous structure. For example, insects, spiders, and flowers preserved in Baltic amber from approximately around 40 million years ago. Then subfossils. These are the remains of organisms that are only partially fossilized and not completely mineralized, often from recent geological periods like Holocene. Okay, these are basically not so old. Okay, these are partially fossilized and not completely mineralized. Okay, found in peat bogs or tar pits or caves, preserving DNA and organic molecules for molecular studies. For example, subfossil lemur bones of lemur bones from Madagascar caves. These bones were found from the Madagascar caves. So, with the help of fossils, how can we determine the age of fossil? Okay, so the method used to determine the age of any fossil is dating method. Okay, this is widely used method to determine the fossil age. It is of two types. First is relative dating and second is absolute dating. In relative dating, this is the method of determining the chronological sequence of fossils and rock layers without assigning a numerical age. Okay, it is mainly based on stratigraphic principle and the use of index fossil. Basically, two terms we have to remember. First, it determines the chronological sequence of fossils and rock layers. Okay, and second, it is based it is based upon it is based on stratigraphic method. Okay, so what is the process of relative dating? The first is the law of superposition. Okay, according to this law, in undisturbed sedimentary rocks, the oldest layer lie at the bottom and the youngest at the top. Okay, here you can see this particular law of superposition says that the oldest rocks or the oldest layer will lie at the bottom and the youngest will be at the top. Okay, the fossils in lower strata are therefore older than those above. Okay, like let us consider this layer as younger and this is older one at the bottom. So first type of fossils are present here and other are present in the bottom layer. So the fossils which are present in the top layer are younger than the present below. Clear? For example, trilobites in deeper Paleozoic rocks are older than the ammonites in overlying Mesozoic layers. Clear? This is the law of superposition, which says that the oldest layer will lie at the bottom and the fossils which are present at the bottom are older than those which are present on the top layer, in the top layer. Okay, second principle is principle of faunal succession. According to it, the fossil organisms succeed one another in a definite recognizable order. Once a sequence is established, fossils can be used to identify relative ages of strata globally. Okay, once uh, we identified a sequence, okay, so these fossils can be easily identified relative to the strata. Like the repetition of ammonites in different Mesozoic deposit confirms their age correlation. Clear? The repetition of ammonites in different Mesozoic deposits or layers confirm their age correlation. Okay. Then index fossils. Certain species lived for a short geological period but were widespread. okay, which were present for a short period, but in that short period they become widespread. Their presence provide a precise marker for the age of the strata. For example, the ammonites in the Mesozoic, trilobites in Paleozoic, and nummulites in Eocene. Clear? Then principle of cross-cutting relationships. Any geological feature like fault, intrusion and dike that cut across a rock layer must be younger than the layer it cuts. Okay. For example, a volcanic intrusion through sedimentary rocks is younger than the fossil in those sediments. Okay, the intrusion is much older than the fossils present in the sediments. Clear? The intrusion, basically the geological feature, it can be any intrusion or it can be faulted rock. Okay, these type of geological features are much older. Sorry, uh, according to this principle, the geological features is younger, not older. Okay, these the volcanic intrusion through sedimentary rocks is younger than the fossils in those sediments. Means these intrusions are younger or developed later. Okay, the fossils are older. Clear? Then correlation of strata. Rock layers from different locations are matched using their fossil content and lithology. This allows reconstruction of continuous relative timelines across large regions. Clear? For example, Glossopteris fossil link coal beds of India, Africa and South America. Evidences for Gondwana. Okay. Lithology is basically the study of physical characteristics of rocks. Okay. It says that any fossil is associated with reconstruction of continuous relative timelines and across the large region. It means they were present in they were widely spread. Okay, and their widespread evidence gives us a proof of the existence of Gondwana. Simply we can say the relative dating is the sequence determination, which means older versus younger. Then it relies on superposition, faunal succession, index fossil, cross-cutting relations and correlation. Okay, we basically relate it with some other factor. Okay, it forms chronological framework into which absolute dating method insert actual ages. Then the second method is absolute dating. Absolute dating basically determine the numerical age. Okay, relative dating basically provides the sequential arrangement, while the absolute dating determines the numerical age of fossils or rocks using natural processes like radioactive decay. Unlike relative dating, it provides exact time frame in years. Okay, so it it is based on the principle of radioactive decay. It means unstable isotopes decay into stable daughter isotopes at a constant measurable rate, which is half-life. Clear? The unstable isotopes decay into stable daughter isotopes at a constant measurable rate, which is known as the half-life. By measuring the ratio of parent to daughter isotope, the age of a rock or fossil can be calculating. Okay, when we compare the ratio of the parent to the daughter isotope, the age of the rock can be calculated. Here you can see, suppose this is the living organism which absorbed C14 radiocarbon during their lifetime. Okay, and when they become fossil, when they become fossil, a small piece of the fossil is burned and converted to carbon dioxide gas. Clear? Stable C12 and unstable C14. Now the unstable isotope will decay into its stable form. So the C14 decays into its daughter N14 and emitting an electron. N14 here is nitrogen and here is the electron emitted. So a radiation counter records the number of electron emitted. In this way, by comparing the ratio between the parent and the daughter isotope, we can determine the age of any fossil. Clear? Here are the different types of absolute dating. The first is radiometric dating. Its principle is radioactive isotopes decay into stable daughter isotopes at a constant half-life. Okay, material dated by this method is rocks, basically minerals and organic remains like C14. Okay, this method is used for the age range of up to 50,000 years. Clear? This is age range of C14, and for potassium argon, millions to billion years we can identify. For example, potassium argon dating of East African hominins fossil. Okay, its limitation is it requires suitable isotopes. Contamination can affect the accuracy. Then there is luminescence dating. The trapped electrons in quartz are released as light when heated or exposed to light. This is the basic principle of this type of dating. The trapped electrons in the quartz are released as light when heated or exposed to light. Basically, sediments, pottery or burnt flints are we can use these materials for dating. Age range from 1000 to 5 lakh years. For example, dating of Paleolithic stone tools in Africa and Asia. Limitation is this method is sensitive to sunlight or heat exposure. Okay, then electron spin resonance. It measures the trapped electron crystal defects using magnetic resonance. These materials can be dated: tooth, enamel, shells, corals and bones. Age range from 50,000 to 1 million years. Okay, for example, ESR dating of Neanderthal and early human teeth in Europe. This requires careful dose rate estimation. Error margin can be large in this method. Then paleomagnetic dating. The magnetic mineral align with Earth's magnetic field and correlated with geomagnetic polarity timescale. Okay, these are the material which can be dated using this method. Time range from 10,000 years to several million of years. Okay, for example, dating East African hominin site using volcanic ash plus the polarity reversal method. It is less precise and work best with radiometric calibration. So here is a sample question that archaeologists found a piece of charcoal, okay, in an ancient fire pit and determined that it contains 25% of its original amount of C14. Okay, the original amount is 25, the remain is 25% of the original amount of C14. If the half-life of C14 is approximately 5730 years, estimate the age of the charcoal. We have to find out the age of the charcoal. Firstly, let's write the given data. So the given data is the remaining C14 is 25% of original, right? Then this means the 75% is decayed, right? If only 25% is present, then it means 75% is decayed, and the half-life of carbon is given 5730 years. Okay, so as we know, after first half-life 50% remains. Okay, let's suppose original amount of C14 was 100% when after the first half-life it remains 50% only, and after the second half-life it remains only 25%. The half of 50 is 25%, so the remaining amount is 25%, and it means there were two half-lives. Okay, now we have to calculate the age. So the formula for calculating the age of the number of half-life into half-life. Clear? So there were two half-lives, first half-life and the second half-life. So the number of half-life is 2 and the half-life is 5730. So the age of the fossil is 11,460 years. So the age of the charcoal is 11,460 years. Clear? Here is the next question. A volcanic rock was found to contain 75% of argon 40 and 25% of potassium 40. Assuming no argon was originally present and all the argon present now is due to the decay of potassium 40. Calculate the age of the rock. The half-life of potassium 40 is approximately 1.25 billion years. Okay. So here we can write the parent isotope, which is potassium 40. Okay, and daughter isotope, which is argon 40. It is already given that no argon was originally present. All the argon present now is due to the decay of potassium. Means potassium is the parent and it is decayed into the daughter isotope, which is argon 40. Clear? And half-life is given 1.25 billion years. Now, according to it has 75% argon 40 and 25% of potassium 40. So potassium 40 is 25% and argon 40 is 75%. Clear? And we are assuming no organ was originally present. Okay, so the parent is remaining only 25%. Okay, and as we know, after first half life, after first half life, only 50% remains. Clear? And after second half life, 25% remains. Clear? So, for calculating the age of the rock, we have to multiply the number of half lives into t 1 by 2, which is the half life. So, 2 into 1.25, so it will be around 2.5 billion years. This is the age of the rock. Clear? Now, sorry, what are connecting links? As the name suggests, connect means something is connecting the two organisms, right? It is the bridge between two organisms. So the connecting links are the organism that exhibit the characteristics of two different taxonomic groups, thereby bridging the evolutionary gap. Okay, the connecting links are those organisms which have the characters of two different organisms, two different taxonomic groups. They represent the transitional stages in the evolutionary process and provide living or fossil evidence for descent with modification. Clear? Connecting links, unlike fully modern organisms, connecting links retain primitive characters of their ancestor alongside advanced traits of their descendant. It means it shows the parental characters also, and it shows the descendants characters also. Clear? What is the significance? Uh, Basically, they serve as the natural proofs of evolution. These are the natural proof of evolution that this species A, this is species B, and let us suppose this is the connecting link AB. Clear? So this connecting link shows that the species B evolved from the parental or ancestral form. Clear? So this provides the natural proof of evolution, and which confirm the theoretical phylogenetic relationships predicted by the comparative anatomy and embryology. They demonstrate the gradual acquisition of new traits rather than sudden appearance of higher taxa. Clear? They are critical for constructing phylogenetic trees, especially when fossil evidence is incomplete. So, the types of connecting links. First, living connecting links. The organisms which are present today, okay, still existing today, that shows features of two groups. For example, Peripatus onychophora, which connects annelids and arthropods. Please remember all these connecting links, these are important. First is Peripatus, which connect the annelids and the arthropods. What are the annelid features? They have a worm-like body. They have nephridia, which is the excretory organ, and they have segmental muscles. What is the arthropod feature? They have chitinous cuticle, tracheal respiration, they have antennas, and clawed appendages. Okay, so this is a connecting link between the annelids and arthropods. Then balanoglossus of hemichordata, which connects the non-chordates and chordates. The non-chordate traits are it contain the open circulatory system. Okay, then it contains the proboscis, this region, with coelom. Okay, and the chordate features are contain pharyngeal gill slits, dorsal nerve cord. Okay, which is rudimentary in a very primitive form, but it shows chordate characters also, so act as a bridge between the connecting bridge between the non-chordates and the chordates. Then the lungfish, okay, which connect the fishes and the amphibians. Fish traits are have scales, fins, aquatic respiration with gills, and amphibians traits are lungs for aerial respiration, ability to survive in mud during drought. Then duck-billed platypus, which connect the reptile and mammals. Reptilian features are they lays eggs, have cloaca and reptile-like skull. Okay, we are talking about this, and this is lungfish. Mammalian features are fur, mammary glands, and producing milk. These are the mammalian features. Next are fossil connecting links. Okay, also we can call them missing links, which are not present today. Okay, these are the extinct forms. Clear? For example, the Archaeopteryx. Archaeopteryx is a connecting link between reptile and birds. The reptilian traits are teeth in jaws, long tail with vertebra, and clawed fingers. Here you can see the clawed fingers, long tail, clear, and teeth. These are the reptilian features, and avian characters are feathers, wings, and furcula or wishbone. Clear? Then Semoria and Ichthyostega also are the fossil, or we can say the missing connecting links. You can study them. Semoria is the link between amphibian and reptile, and Ichthyostega is between fishes and amphibians. Then, what is the evolutionary value of connecting links? Basically, they validate evolutionary continuity, showing that the higher groups did not arise suddenly but gradually from the ancestral forms. Basically, this is evolution, so it is a proof to evolution. They provide direct evidences to support the comparative morphology, embryology, and molecular data. They are especially important for understanding macroevolutionary transitions such as water to land, like fishes, from fishes to amphibians and reptiles to birds. Clear? Here is the link. Here, these organisms are the connecting links. Here, their traits, their advanced traits, and their significance. You can uh, study this table. Clear? Next, second type of evidences from comparative anatomy and morphology. So, this type of evidences reveal the structural similarities and the differences between the organism. Okay, that can only be explained by the common ancestry and evolutionary divergence. By studying the body parts, organs, and external forms, we can trace the evolutionary relationships between the organisms. Clear? So, we are studying the first type of organs, which are homologous. Clear? Homologous, homo means same. Homo means same. Basically, homologous organs are those organs which have same structure but different function. They have same structure but different function. Okay, They are not necessarily similar in appearance but reveal underlying structural unity, proving divergent evolution. Okay, homologous organ says that let's suppose here is species 1, 2, 3 and 4. All of these have homologous organ and these all species are derived from a common ancestor. Okay, as these are homologous organ means the organs are structurally similar but functionally different. Okay, so the structure is same, it means they uh, evolved from a common ancestor. For example, we'll study in the next slide. Homology arises when organisms inherit the same structural framework from a common ancestor. Clear? Over evolutionary time, these structures are modified due to natural selection, adaptation, and ecological pressure, leading to different functions. Clear? Then the examples of homologous organ. The vertebrate forelimbs is a classical example of homologous organ. The human hand, bat wing, whale flipper, horse foreleg, frog forelimb, these all organs are in homology. Basically, their structure is like that. They have a humerus. This is the humerus. This is the humerus. 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 Then the radio and ulna. This is the radio and ulna. Radio and ulna. Radio and ulna. And radio and ulna. Then the carpals are there, then metacarpals are there, and then phalanges are there. Okay, the basic structural pattern is same in all these taxonomic groups, but they perform different function. Clear? Like man have different function, the horse use the leg for walking and running, bat use it different way, and bird also use it for flying, right? It demonstrates the divergence from a pentadactyl limb ancestor. Clear? It shows that all these species are evolved from a pentadactyl limb ancestor. Clear? Then the mouth parts of insects like mosquito, butterfly, housefly, honeybee, these all have a common plan, right? Structural plan, but they have different function, right? The mosquito mouth parts are piercing and sucking type, butterfly mouth part is siphoning type, housefly sponging type, honeybee chewing and lapping type, but their structure is same. Labrum, mandibles, maxilla, labium, all are there in all these organisms, but they are modified for different functions. Clear? According to their diets. What is the evolutionary significance? First, they provide the evidences for the common ancestry. Clear? And they also provide the proof for divergent evolution, that all the different species with homologous organ are evolved from a common ancestor, and predictive value in phylogeny. Okay, they construct evolutionary relationships and phylogenetic trees. Okay, uh, the information from homologous organ are used in systematics and cladistics to construct the evolutionary relationships and phylogenetic trees. Then the analogous organ. Analogous means different, right? Different. Now, please remember, firstly, same in case of homologous and analogous, as I have written over here. Homo means same. Firstly, we are discussing about the structure, then the function. Same. Ana means different, so different structure but same function. Clear? They arise through convergent evolution. It means all these species all these species have adapted themselves according to the environmental pressure, and shows the convergent evolution. This type of evolution is convergent evolution, right? Homologous organ shows divergent evolution. This was divergent evolution. Here are, these are the diverging rays, okay? Divergent evolution. For example, the wings of birds, bats and insects, okay, they are structurally different but functionally same. Birds also use wings to fly, bats also use fly, insects like butterflies also use wings to fly, but the structure is different. Here is the forewing, here is the hind wing, okay, here is the wing membrane in pterodactyl, radius, humerus is there, humerus, radius. So the structure of all these organs are different in different species, but they perform similar function. Clear? So all these organisms used this organ for flight, but structure and origin are entirely different. Then the eyes of octopus and vertebrates. Both have a camera-like eyes with lens, retina and iris. Clear? Vertebrate eyes develop from neural ectoderm, octopus eyes develop from epidermis. Okay, the function is same, uh, which help them in vision, but they arise from different layers. Like vertebrates eyes are developed from the neural ectoderm, while the octopus eyes are developed from the epidermis. Okay. Then the vestigial organs. Vestigial organs are basically those organs which are functionless in the current generation, but they have a function in their ancestral forms, right? They don't have any function now, but they were functional in the ancestral species. So we can say these were the evolutionary remnants providing direct evidences of descent with modification. In evolutionary history, when organisms adapt to new habitats or lifestyles, some organs become redundant. Okay, natural selection no longer maintain their full development, leading to reduction or loss of function. Vestigiality reflect the evolutionary baggage retained from ancestors. Okay, so these features are uh, retained in the current species. Okay, and were functional in the ancestral form, right? For example, in humans, appendix is there, okay, which is the remnant of large cecum used for cellular digestion in the ancestral form. Now we cannot digest cellulose, that's why it is non-functional. Okay, wisdom teeth are there. Okay, the tailbone is there, which is functional in the primates. Then the nictitating membrane, which is present in the eye. Okay, then the auricular muscles of the ear. All these are examples of vestigial organ. Other in other organisms like hind limb bones in whales and snakes, which is the evidences of terrestrial ancestry. Then the wings of flightless birds, which is retained but adapted to running, swimming and rather than flight. Clear? Same in case of pelvic bones in some flightless insects. Then the atavism. Atavism is basically the reappearance of ancestral characters in the modern organism that had disappeared generations ago. Okay, uh, like the organs which were disappeared a generation ago reappeared in the current generation. It results from the reactivation of suppressed reactivation of suppressed or dormant genes that control those ancestral traits. Unlike vestigial organs, uh, atavistic traits appear occasionally in a few individuals due to genetic development or mutational causes. For example, uh, in rare cases of newborn babies, well, it means baby is born with a small tail, okay, supernumerary nipples, then dense body hair, large canine. All these are example of atavism. Clear? So that's all for today's lecture. This is the part 1. In the part 2 we will discuss the remaining 3 types of evidences: comparative embryology, biogeographical evidences, biochemical and molecular evidences. So that's all for today's lecture. Thank you so much. And if you want any kind of study material or you want this presentation, you can mail us at official.learnsaffair.11@gmail.com. Thank you so much.